At some point in our lives, we've all had a bacterial infection that our body could not fight off on its own. To get better, we went to the doctor, who prescribed antibiotics to kill the bad bacteria. Unfortunately, antibiotics don't always kill 100% of the harmful bacteria. If a few manage to survive, they can go on to spread their resistance, and eventually the antibiotics no longer work. As this happens repeatedly, we create multidrug resistant bacteria, which are also known as superbugs. Superbugs cause millions of infections each year and are projected to cause more than 10 million deaths annually by the year 2050. The fundamental problem with fighting bacteria is that they evolve resistance far faster than we can develop new drugs. My research addresses this gap by developing vaccines effective against multi-drug resistant bacteria. Vaccines work by taking small pieces of pathogens called antigens and presenting them to special immune cells. These memory B and T cells learn to recognize the antigen so that the next time they see the pathogen, they can kill the bacteria before it causes a serious infection. The superbug that I'm targeting is called Acinetobacter baumannii, or AB. It is a leading cause of pneumonia, especially among individuals who are on ventilators, which includes thousands of COVID patients over the past 3 years. It can also cause a catastrophic multi-organ failure of the immune system called sepsis, which often leads to death. My vaccine uses an antigen from Acinetobacter baumannii called an outer membrane vesicle, or OMV. OMVs are small round sacs that are filled with bacterial cargo, like DNA, toxins, or proteins. Since OMVs are extremely diverse, I coat OMV membranes onto the surface of gold nanoparticle cores, which provides additional stability and consistency to my vaccine. Using nanoparticles as my vaccine core also allows me to precisely tailor the size of my vaccine to the preferred range for immune cells. In the same way that excessively long sentences are hard to read when studying for a test, extremely tiny or massive antigens are not preferred when teaching immune cells to recognize and kill bacteria. Once my vaccine is formulated, I immunize mice and then test if they are protected from bacteria. My ABNP vaccine generates a robust antibody response that is specific for AB. It also provokes a rapid immune cell response among a group of first responders that traffic to the site of infection to clear the bacteria, completely protecting the mice from both pneumonia and sepsis, and demonstrating the efficacy of nanoparticle technology in antibacterial vaccines. The best part of the vaccine is that because the nanoparticle core is both safe and stable, we can swap out the bacterial antigen to target other species, rapidly creating new vaccines on demand. For example, I have recently begun testing a new vaccine against another superbug called Pseudomonas aeruginosa, which causes pneumonia in patients similar to AB. Another benefit of using nanotechnology is the ability to add immune boosters, like adjuvants, to increase the efficacy of the vaccine. Multidrug resistant bacterial infections are an increasing threat to human health. Drug discovery cannot outcompete bacterial evolution. We must look for alternative solutions. My research fills the therapeutic gap by providing a platform for the rapid development of new nanoparticle vaccines that are safe and effective against multidrug resistant bacteria.